Please welcome Dr. Goodrich is currently a postdoctoral research fellow at the University of Michigan School of Public Health with research interests at the nexus of toxicology, epigenetics, and environmental epidemiology. Following undergraduate training in cellular and molecular biology, Dr. Goodrich obtained her doctorate in toxicology from the University of Michigan. She received the Michigan Institute for Clinical and Health Research Postdoctoral Translational Scholars Programs Award in 2012 to 2015 to expand her pilot research investigating the epigenetic impact of mercury exposure and its implication for cardiovascular health. In a study population of dental professionals with exposures to inorganic mercury from dental amalgams and methyl mercury from fish consumption, she examines the relationship between mercury, DNA methylation, and cardiometabolic outcomes. Please welcome to the podium, Dr. Goodrich.
Can you hear me now? All right, great. So first off, I'd like to say thanks to, first I have to read this disclaimer. So I do not have any financial interests of any products in my talk or with any companies that are offering grant monies for this educational program today. I'd like to say thanks to Rich Fisher and Jack Callan for inviting me today to speak to you. I've done a lot of work on different kinds of mercury exposures in human populations. And in the past, I did a lot of work on mercury and genetics, which Dr. Ashner talked about. So if you're interested in that sort of thing, just find me at some point during the conference and we can chat. But I've heard that a lot of people here have a great interest in learning about epigenetics, which is a up-and-coming thing in the environmental health field especially. So today, I'll be telling you specifically about epigenetics. First, just giving a broad background on what it is, because I'm sure some people know all about it, some people know nothing about it, and everywhere in between. So I'd like to just give you a background on what it is and why it's important for health research, and then get into what we know about mercury and epigenetic change so far. So epigenetics was actually first defined quite a while ago in 1942 by a scientist called Conrad Waddington as the branch of biology which studies the interactions of genes which bring genes with the environment which bring their phenotype into being. And some of the thought of this is that in every cell, you can think of it as this marble at the top here, you actually have the same genome. So in every cell in your body, you have the same genetic code with the exception of maybe a few mutations here and there. But a stem cell can turn into a liver cell, a cardiac myocyte, a neuron, and all sorts of different things, even though they have the same exact genome. So there are many different things that cause those differences and that differentiation, but a large part of that we now know is the epigenome. And the epigenome, the word literally means over above the genome. So it's something on top of, in addition to the genetic sequence that affects how genes are expressed and ultimately phenotype. And the kind of longer, more scientific definition that we use for it now are mitotically and or meiotically heritable changes in gene expression that do not involve changes to the DNA sequence. So in mitosis, when your cells divide, the new cell is going to have the same genetic sequence as the old cell. But the thought is that when a neuron divides, the new neuron also has the same epigenome as the old neuron, and thus it can remain a neuron. Whereas opposed to a different kind of cell has an epigenome more related to that cell type. But even though these are meiotically and mitotically heritable, they can also change due to environmental exposures and things like that, and over the course of a lifespan of an individual. And I'll get into that because that is where that's what's more of interest to health and medical research. But first, I'd like to tell you about the major kinds of epigenetic modifications and what they're doing. So when we think about DNA, all of the DNA in each cell, if you take all of your DNA and you stretched it out super thin and super straight, it would actually be about 1 meter long. But you have to fit, that DNA that in theory is 1 meter long into a cell that's only a micrometer long and into the nucleus, which is even smaller. So the body does that by DNA is actually packaged into chromosomes, which are much, much smaller. And these chromosomes have a very exquisite way in which DNA is wrapped around nucleosome proteins, and then they are condensed with one another, ultimately to form this structure. But it's not a haphazard structure. It's very well regulated, and it's done so in large part by these proteins that are involved in nucleosome formation, as well as these epigenetic modifications. So this is a histone protein here that DNA is wrapped around, but histones can be modified. There are many different kinds of modifications that are done on the tails of histones, which ultimately affect how closely in a given region of the chromosome these nucleosomes are going to be packed together. And in really packed together areas, which we often call a heterochromatin, the genes there can't really be expressed. Transcription factors can't get in. You can't really have gene expression there. But if you have certain other kinds of histone modifications, they'll make that region of the chromosome more what we call loose. Or euchromatin. So it'll be better accessed by transcription factors. Genes can be transcribed there and things like that. Likewise, there are certain kinds of non-coding RNAs that are involved in creating this repressive structure. And DNA methylation is more what I study, so I'll talk about it more later, but this is a modification that's actually directly on the DNA, not on those histone proteins, and it involves methylating a cysteine residue. So cysteine is one of the 4 bases that make up your DNA, ACGT, and in mammalian species, Cs that are upstream of G, so sequences that are CG, and then whatever else, can be methylated. And when it's methylated, genes are often not expressed, and when it's not methylated, it's more likely for them to be expressed. And that modification is probably the easiest to study, so it's the most well-studied right now, and it's also what I'm studying. But these other kinds of epigenetic modifications are also very important in gene expression and can be changed by environmental exposures. So this is just another picture kind of depicting the same thing. When we think about gene expression, which is ultimately what brings about phenotypes, it of course depends on our genetic sequence, but it also depends on these epigenetic modifications such as DNA methylation and the histone code, which will vary by cell type, it'll vary by disease state, it will vary if you're exposed to different things, and that's why we find it to be a very important field right now. And this is just another picture too to show that everything in your body started from a stem cell at some point in time, and they turned into all these different cell types. And so the genetic content, once again, is the same in all those cell types, but the epigenome is quite different. And much like these epigenetic changes can lead to differentiation of cell types, it can also contribute to adverse health outcomes. So it's well known in cancer that the epigenome in cancerous cells is much, much different than that in healthy cells. It's been observed to contribute to chronic inflammation, to many other things that are not listed here. They're basically epigenetic changes that have been associated with most of the diseases you could possibly think of that have been studied to date. Another way to think about epigenetics and how it contributes to disease is to think about twins. So identical twins, through their whole life, they have basically the same genetic code or exactly the same genetic code. When they're born, they have basically the same epigenome as well. But even so, when one twin has asthma or develops cardiovascular disease or schizophrenia when they're adults, the other twin does not necessarily always develop it. So we know that environmental factors contribute to all of these diseases, not just genetics because of that, but epigenetics also may be the link between the environmental exposures and the disease later in life. And this is there was a study done about 10 years ago that compared the epigenome of young twins and also old identical twins, older. So this is 2 chromosomes from 3-year-old twins. This is one twin, this is another twin. Basically, if you see yellow here, it means that the epigenetic marks that they're studying with this method are basically the same. And if you see red or green, it means it's different between the twins. And so you can see that at 3 years of age, their epigenomes across all the sets of twins that they studied were very, very similar. But by the time they reached midlife, their epigenomes were very different. And this is because, the twins went off, did different things, had different exposures, different lifestyles, and that changed their epigenome. This can also contribute to the changes in disease development that identical twins experience. And sorry, this might be a little bit hard to see, but what are some of those environmental factors that are impacting the epigenome across the lifespan? And this one here lists just chemicals, but some of the ones that have been studied to date that have been shown to impact the epigenome are endocrine disruptors such as bisphenol A, phthalates, heavy metals, which is what I study mostly, such as mercury and lead, in which I'll talk about mercury later, air pollution, particulate matter, pesticides, but there are also nutritional factors that can modify the genome for a lot of times in a good way, and in some ways we're not sure if it's a good way or not yet. There are also stressors, being exposed to stress, such as violence, maternal stress during pregnancy, things like that can lead to epigenetic changes. And as far as the chemicals go, we know mechanistically, broadly, how this can happen. So there are different ways depending on the chemical, but some chemicals alter the expression or the actual function of particular genes that are involved in maintaining those epigenetic marks. So when a cell divides, the DNA methylation has to end up in the new cell like it was in the old cell. So there are certain enzymes called DNA methyltransferases that make sure that happens. So some chemicals can inhibit these enzymes, which throws off the epigenome in the new cell. Some chemicals can also disrupt pathways like the 1-carbon metabolism pathway, and this pathway is important for many, many things, and some of you probably know about it for different reasons. But as far as DNA methylation and in some cases histone methylation goes, it's ultimately the provider of the methyl group that you need for that methylation event. And when it comes down to it, what we're interested in doing in this field now is, since we know some of these toxicants can alter some of these pathways, and we've seen that they've been associated with changes to epigenetic marks, are these changes linked to short-term or long-term and latent health outcomes that we know are associated with these exposures? And before I get into mercury, there are a couple of other things that are really important in epigenetic research in general, which I think you might be interested in knowing about. So one aspect is the timing of exposures. So does it matter? Does it matter if you're exposed to mercury or some other chemical at any point in time, or are certain time periods more important for epigenetic change? And the answer is that all time points, you can have epigenetic change at any time point from exposure, but some time points may be a little more important and susceptible than others. So basically, one extremely important time point is right around conception, in those very, very early days as the embryo is growing and dividing. There is a widespread erasure of the maternal and paternal epigenome and a reestablishment of the offspring's epigenome at that time. And when this happens, you're establishing the epigenome that can essentially propagate to all the cells that the offspring will continue to divide into. And some of these marks can remain for their entire life. So if you have an exposure at this particular time, then it could have a big impact on all the cells that divide from then on out if it causes some kind of aberrant epigenetic change. But even though that's a very, very important time point, and I'll focus on that time point for a little bit throughout the presentation, these other time points have been shown to be important too. myself and others have studied exposures during early childhood, during adolescence, during adulthood, and shown that different exposures, stressors, lifestyle factors can influence epigenetic propagation and gene expression and things like that later in life too, not just at this early developmental time point. So if you're exposed to something early on, it doesn't mean, it may unfortunately negatively impact your epigenome and your health later, but fortunately there are other time points of exposure that are important too. So you're not set, as you know, your genetic code is set. Once you're born, that's what it's going to be for the rest of your life, but your epigenome is plastic, can change to some extent. And in animal studies, we have the ability to study the epigenome in any tissue type. So if you're interested in toxicity in the brain, you can study the epigenetic marks specifically in the brain. But most of my work is done in humans, and of course, we can't just obtain brains and lungs and other tissues like that from live subjects, fortunately. So we study instead peripheral tissues that we can collect. So when we're looking at prenatal exposures, once in a while you can get genetic material from amniocentesis. That's actually kind of hard to get in studies too, but a lot of studies collect placenta, umbilical cord blood, sometimes maternal blood during pregnancy to start to look at these marks. When we're studying children, adolescents, adults, the most common samples collected are blood and saliva that we look at epigenetic in changes in these tissues, once in a while hair, stool, things like that. And so the thing to keep in mind is that most of the studies I'll be talking about, we look at the epigenome in blood or saliva, but we aren't able to access tissues of interest such as the brain. So it's possible that we're seeing similar epigenetic changes in the target tissues from these exposures, or potentially even worse epigenetic changes since the exposures accumulate there, but we cannot say that for sure at this time. So for a couple of minutes, I'd like to also talk about something that's important to the field of epigenetics, the developmental origins of health and disease, what we call the DOHAD hypothesis. So this came about many decades ago by a scientist with the last name of Barker. So it used to be called the Barker hypothesis. And he was finding that the early in utero environment seem to be associated with changes much later in life. So, for instance, babies who were born with low birth weight, when they became older, they were more likely to develop obesity, diabetes, things like that. And, so you may wonder, how does such an early change influence the health outcomes much, much later in life? And we started to learn that epigenetic change is one of the ways that this is occurring. So I talked earlier in the timing slide about how exposures early on can influence the epigenome potentially for the rest of the subject's entire life. And that plays into this DOHAD hypothesis. So in early development, if certain exposures modify the genome, the epigenome that's carried through the life of this individual, and then certain modifiers might impact it more later in life too, and ultimately these changes will contribute to the phenotype in the adult. Some changes might contribute to adverse health outcomes later in life. Some changes might actually make an individual healthier later in life. And some of the ways broadly that this can occur is that environmental chemicals, nutritional differences during pregnancy that the mother is exposed to can alter hormonal regulation, metabolic pathways, oxidative stress, things like that, which can impact that epigenetic programming in the offspring. And this, if it gets carried on as the offspring grows, can lead to long-term effects on gene expression, which may alter the disease risk. And we've been able to study this DOHAD hypothesis pretty well in a very neat mouse model called the agouti mouse model. So you wouldn't guess it by looking at it, but these 5 mice here are all genetically identical. But you can see by looking at their fur that they're very different. And this is all due to an epigenetic difference in these mice. So these particular mice have a very epigenetically labile locus, or one that can be very different between individuals. And in mice that end up looking yellow, at this locus, they have epigenetic modifications that lead to heavy expression of this agouti gene. And this agouti gene, we don't really want it to be expressed, as it turns out. It leads to many adverse outcomes. These mice, when they're older, end up, these are young mice here, but when they're older, the yellow mice end up more obese. They have a greater propensity to develop tumors and things like that. While these brown mice are the healthy version. They stay lean, even though they're eating the same food as the yellow mice. They don't really develop tumors, things like that. And this is all due to just an epigenetic difference as this one particular locus. And so in a normal mouse population, it's kind of what we call stochastically regulated. So early in development, some mice are just more methylated there and some mice just get less methylated there. And you end up with this distribution of most mice kind of having this middle ground phenotype, some being more yellow, some being more brown and healthy. But what was discovered by actually the scientist who I work with now, Dana Dolinoy, and her collaborators at the time, was that certain exposures can alter the distribution of the offspring at this gene. So one of the exposures they used was bisphenol A, which some of you might know about. It's actually sometimes used in certain kind of dental composites, but also used to be in a lot of plastic products, and it's still used in receipt paper and some can linings and things like that. So basically they found that if the mothers were exposed to BPA throughout the entirety of pregnancy and before and for a short time afterwards as well, the more offspring ended up being less methylated and having this yellow, more prone to obesity, more prone to tumors phenotype than would have normally occurred without the exposure. And what was really neat, Dana also did an experiment where they tried to combat this effect and gave the offspring bisphenol A, but at the same time, they tried a couple of different methyl donors. One was genistein, which is found in soy. And what happened is that the distribution came back to normal, what it usually is in an unexposed mouse population. So in many ways, being exposed to something bad and essentially something good at the same time was able to bring these mice essentially back to normal. And so that was done with bisphenol A, but there are different kinds of exposures that have been shown to do something similar. We haven't used this model yet to study mercury, but that would definitely be neat to do in the future if we can get the funding and whatnot to do so. So in general, before we get into mercury, and I just want to give, kind of broadly again, reiterate the importance of this doe-head aspect of epigenetics, even though I'm not just going to talk about doe-head and the mercury studies, but you might hear about it when you're hearing about epigenetics. So these prenatal exposures can alter epigenetic programming that will cause changes to gene expression that can propagate throughout the offspring's life. This can lead to developmental impacts early on, developmental changes and growth and things like that, and ultimately susceptibility to adult disease. And fortunately, I don't have to give much of a background on mercury because Dr. Ashner did a great job of that already, and I'm sure a lot of you know about it already as well. But I'll also be talking about not the three forms, but two different forms of mercury today. So it's nice that you've heard about both of them. And I'll just, kind of give a couple quick liners on them again as well to remind you why we care about different forms of mercury. The U.S. Environmental Protection Agency cares a lot about mercury. They rank it as the top 3 priority pollutant, mostly because of mercury in fish. So because it's in fish and the public can't help that, but we don't want to tell the public you can't eat fish, it's a problem, and it's something that we're trying to slowly work towards cleaning up in the environment. But as you heard in the last presentation, that's a bit hard to do, even though we reduced our industrial standards so that coal-fired power plants are required to emit less mercury now into the atmosphere in the US. It's still a problem around the world. Places like India and China, where industry and populations are growing, there's a lot of mercury emission there, and because it travels so far in the atmosphere, it can end up in other parts of the world as well. And of course, it's mercury. Other forms of mercury are in other products important to health, as you know. It's still used in the silver dental fillings, and there's always, a lot of debate about whether or not those should be used. And then occupational exposures are very important. Of course, the dentists and dental hygienists and assistants that use mercury fillings are exposed, but there are certain it's kind of hard to tell from this picture, but there are certain gold mining communities around the world, mostly in developing nations, that use elemental mercury to mine gold, basically because it binds to gold. And then what they do is when they have the sediment with mercury bound to gold, and then they burn off the mercury and they have their gold. But they're basically, most of the time, they're right there when the mercury burning is occurring, so they're inhaling tons and tons of mercury vapor. And there are certain other occupational groups that have exposure as well. So mercury's, as we know, still out there, still an important public health concern, and there's always debate about what the safe levels are. Of course, probably most of us here would like to say no mercury is the safest way to go, but the regulatory agencies are always kind of going back and forth on that. But one thing we know about mercury, that of course that there's genetic susceptibility. Some individuals are more susceptible than others, but also that it can have latent health outcomes. You might be exposed today or for a couple of decades and then see the health outcomes, toxicity from it later on. So how is this occurring? We know a lot about many different kinds of mechanisms related to mercury toxicity, some about what you heard about in the last presentation, but we also think that epigenetic change is one mechanism that could be contributing to mercury, especially those latent effects we see from early exposures and outcomes later on. And you've already heard about this, but I'll be talking about basically two major species of mercury as well. Mercury vapor, in the populations I'll be talking about, the main exposure is just going to be from dental amalgams. I'm not going to talk about any minors today. When we study mercury vapor in human populations, a great biomarker to use is urine. A lot of this mercury is associated with urinary levels. Methylmercury is that form that's you're exposed to from fish consumption. And when studying this species in humans, blood and hair are often great biomarkers to use. The levels there are highly associated with fish consumption of the subjects. So I'll be using all of those biomarkers in my studies. When we start to think about mercury and epigenetics, why should we study this at all? So of course, there's that thought that the latent health effects might be underlain by epigenetic change, but another reason is that we know mercury can potentially disrupt epigenetic processes. So several, mostly in vitro studies, some in vivo studies, this is a simple version of the 1-carbon metabolism pathway that impacts DNA methylation. So here, basically everything that's going on here leads to donation of a methyl group that can then be used to methylate CpG in DNA. And everywhere where there's a mercury box are areas of this pathway that mercury has been shown to disrupt in some species or some in vitro model or another. So it can directly inhibit a couple of enzymes that are important in this pathway. It can interfere with the homocysteine balance. It binds up a lot of glutathione, things like that. So basically, mercury has been shown to impact this pathway. And if it impacts this pathway, it probably impacts DNA methylation. So you can't really read this, probably, but I'll explain it a little bit. We made this table about 2 years ago, and at that time, these were essentially all of the studies looking at mercury and DNA methylation, and a couple with histone modifications. And at that time, evidence was starting to grow that mercury can impact epigenetic markers. A lot of it was done by Neil Bosu and his colleagues at McGill, formerly at University of Michigan, looking at species, different kinds of animal species, polar bear, mink, kind of unusual animal species, but that have high methylmercury exposure from fish consumption. And they saw that there were changes to DNA methylation patterns kind of globally from being exposed to high levels of methylmercury. There have been some lab studies looking at mice, rats, earthworms that start to show some epigenetic changes. Most of these have been done with methylmercury, though a few have been done with inorganic species as well, which is probably more of interest to everyone here. And then at that time, only a handful of human studies had been done, and they found associations between methylation at a gene here or there and these mercury levels. So at that time, myself and my research group thought this was a good time to start studying this ourselves. But I guess I wanted to talk about this before I get into that. So I've actually only studied mercury and epigenetic change more cross-sectionally in adult populations. But since I told you a lot about DOHAD, I wanted to tell you very briefly what has been done with mercury and DOHAD in the last couple of years as well. So once again, I haven't studied these development exposures yet myself with mercury, but there have been a couple of rodent studies and now 2 very, very recent human studies that have done so. And they've definitely found epigenetic change linked to prenatal exposures. To me, the most interesting study was a study performed in mice that used a similar exposure paradigm that we use with the agouti mice, where they exposed the mother before she became pregnant, the whole time she was pregnant, and then throughout weaning, and then stopped the exposure. So the offspring were only ever exposed through the mother, and then they were never exposed again as they were developing into adults. Even so, at a particular gene that was of interest to this study called BDNF, brain-derived neurotrophic factor, this gene is associated with many different neurological and neurobehavioral outcomes, and it's very, very important for that. They found that this prenatal methylmercury exposure, the white here are mice that were not exposed prenatally, the black are mice that were, and these are different methylated sites at this particular gene. They found that at different time points, right after the exposure and even up to 14 months after the exposure, the mice from the different exposure groups had differential methylation at this gene, and it was associated with differential expression of this gene too. And all of that together, basically in the mice prenatally exposed, they had heavy methylation of this gene that persisted throughout. Basically 14 months is pretty old for a mouse. So it's definitely past midlife for most mice species, but they found all the way through that time point, the methylation changes persisted at this gene, the expression changes persisted at this gene, and these mice also had depressive life symptoms and some other neurobehavioral symptoms from just that early exposure with no other exposure after that. And apart from methylation, they also studied histone code changes, and they saw persistent histone code changes that were also known to be linked to gene repression at this particular gene. So that's in mice. Nothing that profound has been shown or been done in humans yet, but there have been a couple of studies looking at DNA methylation in offspring right when they're born using cord blood DNA and comparing that to the maternal mercury levels that reflect the prenatal exposure. And this, I'll explain this to you, but briefly, in this study, the mothers didn't really have particularly high exposures. It was actually much like you'd see in the general population, not an occupationally exposed group or anything like that. And toenail mercury is mostly a better indicator of that methylmercury exposure that the mothers have. And they looked at a panel that assesses methylation at almost 500,000 CpG sites across the genome, so it gives you a really broad picture of what's going on everywhere. And basically, any dot that you see higher up here, this is the negative log of the p-value, so a higher here means it was more statistically significant. And on the positive here means that it was hypermethylated in the exposed group, negative means it was less methylated in the exposed group. And you could see that by looking at exposure, there weren't a lot of highly significant sites, but there were some, and this was an exposure, like I said, the exposures in this population were actually quite low. And even then they saw some epigenetic changes in the offspring associated with those exposures. And they tended to be in certain kinds of regions too. So when we study methylation, we often talk about CpG islands. CpG islands are CpG dense areas where if they're heavily methylated, you usually see no expression of the gene it's near, and if they're lowly methylated, you see a lot of expression of those genes. And you often find these in promoter regions of genes, so they're pretty important for gene regulation. But we found that the differences, the most differentially methylated sites by mercury exposure were not really in those CpG islands. The gray bar here is all of the sites on this array. The blue bar is what you see enriched in the top sites, differentially methylated by mercury. But instead they're in something called the North Shore, which is, we're now knowing, which is a little bit upstream of CpG islands, and we're finding that these regions are important for regulation as well. In another very recent study just published, I think a couple months ago, that looked at a similar question in another population using another array that looks across much of the epigenome, we found that once again, this study used maternal blood mercury, cord blood mercury, and they measured the total mercury concentration as well as the methylmercury specific concentration, and they found that there were a couple of different genes that were differentially methylated by mercury exposure. This particular one is involved in transcription. It's essentially a transcription factor. And the black line here are the offspring that had the lowest exposure, and the really light gray line are the offspring that had the highest quartile of exposure. And there's this differentially methylated region of this particular gene where it's much more hypomethylated compared to those that are lowly exposed. And the neat thing about this is that sometimes you can see findings like this in a population study, and it could basically just be by chance. In this particular population, something weird was going on and you happen to see that change. But this finding was replicated in 2 different populations. And so I think those researchers who are at Johns Hopkins right now are looking more into this and what this particular gene might mean for development and ultimately DOHAD type outcomes you see with early life methylmercury exposure. So now I'll get into more of my work. While the evidence is growing for early life exposures and how they can impact the epigenome and early life exposures to mercury specifically, I was interested in later in life exposures to mercury and if they're impacting, the epigenome basically now. So the first study I looked at this in was essentially a pilot. I had recruited a group of dental professionals, dentists, hygienist assistants, and a couple of other related occupations at 2 different meetings of the Michigan Dental Association, which is where I'm located in Michigan. And these studies, we originally conducted these studies to look at genetic modifications that influence mercury accumulation, and we found some neat results there. But we wanted to start looking at epigenetics in this population as well. At the time, we didn't really have the money to do a whole across the epigenome study, so I decided to look at a couple of specific candidate genes. And this one gene in particular, our group selected because it's a selenoprotein. Selenoproteins are really neat. They can actually bind mercury exposure, and they can bind mercury, and so in some ways they're protective against mercury exposure. They can bind both the inorganic and the methylmercury form. And also so we looked at the methylation of the promoter of this gene, which could impact the expression of that gene, and found that when we looked at hair mercury levels, which are typically more indicative of those methylmercury exposure, among the males, we saw less methylation with higher mercury exposure, and among the females, didn't see the same thing. And this is something that we actually see a lot in toxicology, gender-specific differences in response to toxicants. But there was a bit of a caveat to this population. Most of the females were dental hygienists and assistants, and they actually had lower exposure than the dentists in the study. So it could have been, an occupational factor thing or something to do with their exposure levels. So also quickly looked at this in just the dentists. These are the male dentists, these are the female dentists, and saw the same thing, only a difference in the males. So this was very interesting, and by looking at basically only 4 genes in this study, I observed one that was significantly associated with mercury exposure. So this led us to want to look at methylation across the epigenome. So I set out to do that by recruiting a new study population from the ADA, or the American Dental Association, which most of you are probably familiar with. And in this study, when we recruited this population, the main aims, there are a couple aims of the study, but it was primarily to identify regions of the epigenome that are modified by methylmercury and elemental mercury, or potentially one or the other, And also in this study, I'm not really going to talk about it today, but I was also interested in looking at if mercury impacts various cardiovascular and metabolic health outcomes, because that's something that's growing in the literature right now. And ultimately, too, to determine if those epigenetic changes are mediating the relationship we see between the mercury and ultimately the health outcomes. And just briefly, in designing this study, we collected 2 biomarkers to use to represent their methylmercury exposure, also collected a lot of information on their specific fish consumption, collected 1 biomarker urine to study elemental mercury exposure. And then, as far as the epigenetic outcomes were concerned, looked at a few candidate genes, some of the ones that we studied in the Michigan Dental Association study, as well as used an epigenome-wide array to study sites across the genome, collected various cardiometabolic outcome measures, which I can talk to you about outside of this if you like, and ultimately we're looking at the mediation between these two as well. So just in brief, I know I've already talked about DNA methylation, but just to give you another reminder of kind of what it is and why it's important, DNA methylation is a direct modification of cytosine bases. And when this methyl group is added to a cytosine, it's often associated with gene repression, and that has to do with basically when it's methylated, a certain protein binds to heavily methylated regions, and then when that happens, transcription factors can't really bind, so the genes there can't really be expressed. And these patterns are typically heritable. When a cell divides, you'll see in the same methylation in the new cell, but it can be impacted by exposures. And when we study this in the lab, this is kind of a really simple schematic to try to show it. We use something called bisulfite sequencing. So there's this compound called sodium bisulfite, which will actually it's very harmful to DNA, but when you treat DNA samples with it, it will convert any cytosine that's not methylated into a uracil, and then when you run that through PCR, it's essentially becomes a thymine. So it essentially converts C's to T's unless they're methylated. If you have a methylated cytosine, it's not converted. So you can use different kind of methods to sequence after this treatment. And if you have a T there, you know it had been unmethylated. If you have a C, you know it was methylated. And essentially that's how we figure that out in the lab. So what was this study population like? We couldn't use them all for the epigenetic analysis, but we had over 600 individuals participate in this study. They were a fairly healthy bunch, more males than females. They were recruited from a meeting in California. They were from all over the US, but a lot of them were from California. We had 2 major racial groups in the study population. We had 65% that identified as Caucasian and 27% that identified as Asian, as well as a smaller amount of other racial groups. They were mostly dentists, so 90% were dentists from the US or elsewhere, and then a smaller proportion were hygienist assistants and a few other occupations. So what did we see with regards to mercury levels in this population? And once again, hair and blood are more representative of that methyl exposure, urine of inorganic. When we looked at their levels, dentists had higher urine levels indicative of that occupational exposure than the hygienists, and this average level of 1.5 in the urine is good, because a couple decades ago when studies were done at meetings like this, dentist levels averaged 20 and things like that. So the good news is that a lot more dentists are using less mercury, or if they're using it, protecting themselves more from it. So occupational exposures are decreasing, which is great, but this is still about 3 times higher than the general average US adult population, even so. And the interesting thing was that their exposures to methylmercury were higher than the general US population as well. And when looking at their fish consumption data specifically, they did consume about 3 times more fish than the average US population does. And this is something that we often see in higher socioeconomic status type classes, like more highly educated people like dentists and doctors and other occupations like that often do eat more fish, because we know fish is healthy. But you do have to also consider what's happening if you're getting this higher mercury exposure along with your fish consumption. And then just briefly, when we compared males versus females, regardless of occupation, males across the board had higher exposure levels than the females. So with regards to the DNA methylation, we set out again to look at that same gene, selenoprotein, that we looked at in the last study. And this time, I was able to use two sources of DNA, since we know the epigenome can differ by tissue type. This, among the same individuals, these are the methylation levels at this gene using blood DNA, and this are the levels using saliva. So you can see, even with these two tissue types, the methylation differences at one gene are very different. And in the epigenetic field, we're now trying to figure out what is the best biomarker to use, because, you know, as I said before, we can only collect things like blood, saliva, once in a while other tissues, but we're trying to figure out what they mean with regards to representing tissues of interest and things like that. But I think for now, the best thing to do, if you can collect more than one tissue, you should do it, because one might be a better biomarker than another, and you're going to see differences between the tissues. So what did we see? We actually didn't replicate that finding in this population with the hair mercury levels and the methylation levels at this gene, but instead saw differences in methylation at this gene in the opposite direction with regards to urine mercury levels, which represent that elemental mercury exposure. And what this means for expression, we'll have to follow up with in another study, because we didn't have RNA, we couldn't get RNA from the samples we collected in this particular study. But moving on, and I don't actually know what time it is, but I'll try to wrap up because I'm probably running over. But I'll tell you very briefly about the epigenome-wide array we used in this population. So I was also able to use that array that looks at methylation at almost 500,000 sites across the epigenome, but it's rather expensive, so we could only look at this in a small subset of the population. While we had up to 600 individuals in this study, we only selected 48 male dentists to study methylation patterns in. And in the future, I'm working on getting the funding to study a female subset as well. And basically, I selected this group to have low and high mercury exposures, to be within the same age group, and to have the same racial distributions. And so what did we find? The biggest differences in methylation were not by race. I'm sorry, were not by exposure, but they were by race. So these are all the Caucasians, and these are all the Asians, and when you use all the data, they cluster mostly by race. So there are a lot of racial differences in the epigenome, much like we see with genetics as well. But what about mercury exposure? After using very robust statistical methods that adjusted for a lot, comparing, doing so many tests, comparing almost 500,000 sites, we found that 32 different sites and 32 different genes were differentially methylated by hair mercury levels. None of these were in genes that I had really thought about before. So now we're trying to figure out what these genes are, what they might be doing with regards to mercury toxicity. And when looking at urine mercury levels, there were only 3, or in a different iteration of the model, 9, that were associated with methylation. So I think methylation studies with inorganic mercury exposure still have a ways to go. Most of the studies to date have been done with methyl mercury exposure. We're seeing more effects from methyl mercury exposure so far, but elemental mercury exposure is also impacting the epigenome, possibly to a lesser extent, and I think we want to continue this area of research to further look into that. And in the bigger picture overall, this is kind of where my current work is going. This is a very busy graph, but ultimately, I'm interested in looking at how, when people are exposed to both kinds of mercury, does it impact DNA methylation? Do those DNA methylation changes impact cardiometabolic risk factors, or ultimately other forms of mercury toxicity as well? And are there certain nutritional factors, such as polyunsaturated fatty acids or selenium, which also come with fish consumption, that can alleviate some of this change, protect against some of this change? And ultimately, there are a lot of other factors that are coming into play to impact these outcomes as well. But in the real human body, we often study one thing at a time, one chemical, one outcome. But in real people, this is what's going on. Complex interactions between many exposures, many lifestyle factors, genetics, epigenetics, and ultimately all of this is leading to disease outcomes. So in summary, hopefully from the beginning of the presentation, you've started to get a handle on what epigenetics is. And if you'd like to know more, just find me and I'd love to chat about it. But as far as mercury and epigenetics goes, we are seeing some effects, mostly with methylmercury, but some with elemental mercury as well, between exposures and DNA methylation. Specifically, I observed a few of these associations in 2 cross-sectional studies of adults. These effects may differ by the tissue type, and what's happening in target tissues of interest, such as brain, kidney, are something that we need to learn more about in the future. In perinatal changes, there is some evidence for DOHAD-like changes, early life changes from maternal exposures that could propagate throughout the individual's life. And while the effect size, the actual biological change that's happening, even when it's statistically significant, it's actually small. And what this means functionally, we're still in the process of learning about. But even though the effect size is small, I care about mercury and methylmercury in these studies here, but these individuals are exposed to, as you know, many other exposures. You know, people are exposed to lead, endocrine disruptors, secondhand smoke, all kinds of things throughout the life course. And the impact of all of these environmental factors on the epigenome over time could be immense. And that's something that is of really big interest in the environmental health field right now. And that's important for cumulative risk assessment. And so in brief, I'd like to acknowledge this is Dana Dolinoy, whose lab I work in currently, who did a lot of the agouti mouse work. And Dana, Al Francebell, and Del Bassi were very instrumental in the American Dental Association and the Michigan Dental Association work, as well as many other individuals who helped out with the lab work there. And we have been generously funded by the NIHS, the Environmental Protection Agency, for various parts of these projects, as well as the NIH Translational Science Branch. And thank you.